Hello, people. Let's see your faces. Only 10 of you here today. Wow. Elijah, hello. Michelle, Vanessa, Rocky, Farouk. Farouk, let's see your face. We never get to see you. I don't even know if the name, there he is. Okay, Farouk. Farouk, you write a lot in your answers. That means I have to read a lot. If you could be a little more focused.
Sorry about
The key, very complete answers. Very complete answers. But the key to doing well in my class is always giving me less work to do. Mairead knows that. She's been through this before. If you can cut Dr. Slish's workload down, an A. There's Alex. Christopher James. Come on, Christopher. Anthony. Anthony Estevez. You know, if you're from Philly, your name would be Anthony. Is that what it is in New York too? I don't know. Definitely not in Plattsburgh. Anthony. Sammy's here. Sammy's here. So a lot of you people don't know this, but Audra Santange, she's spoofing us right now. She's actually in Chuck, but she's got a virtual background of the library, so it looks like she's in the library. But no, she's banging those pleasure machines at Chuck E. Am I right, Audra?
No.
Oh God! Speaking of Chuck E. Cheese, my daughter, she's 10 now. I think when she was about 6, I took her to Chuck E. Cheese, and there's this one machine. You pull down this huge lever, and this huge wheel rolls and rolls, and you get like 2, 3 tickets. She hit the 100 ticket once, and they came pouring out. It was like, oh, it was joy. It was pure joy. And for 100 tickets, you can get, a Tootsie Roll. Okay. Tyler Montano is here. Kwame. I haven't had a semester without Kwame in about 5 years, I think. He's in all my classes. You know what that means, right? That's tough. You know what that means, right, Kwame? I can fail you at any time. You're under my control. I can fail you in this class and you can't graduate. You know I will. All right, well, let's see. What time is it? It must be a tad after 1 at this point. Why don't we go ahead and get started? I'd like to start with going over the last exam, which I think most people did pretty well on it. So I was kind of pleased. I think, I don't know because I don't take roll, but I think showing up to class means a lot because some people just had no idea what I was asking for the questions. And other people were like, oh, yeah, this is what you said. Here you go. So I think it really does help to show up. So anyway, let's do the multiple choice. We usually don't. Hang on. Back.
So, Professor Slish, you sent an email talking about a list or something. Was that for this class
Oh,
or for cell bio?
that was cell biology. So I said list the mechanisms that do this. Now, what does a list mean? It means bang, 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 bang. People are giving me paragraphs this long that I have to read. Like, oh, Jesus. Do you know how many of these I have to do? I'm just being a jerk, though. Okay, match the type of protein, i.e. its function, with the factor that is involved in development. Ooh, this sounds like a good question. Use each one only once. Ectodermin. What is the activity of ectodermin, Mairead? If you were to guess.
Ectodermin is a ubiquitinating enzyme.
I believe so. How about frizzled? Audra? Frizzled. You know, frizzled. Audra, you're not going to help us on this one?
No.
Let's try hormone receptor. All right, I'm sensing that you want me to give you the answers. Let's see if I know them. Disheveled. Disheveled is a signal trans what? Transdiction molecule. BMP is a hormone. Cerberus is a blocking agent. Is that a hormone blocker? There it is. And VEG-T is a transcription factor. Let's see if I'm right. Let's see if I'm right. Oh, it doesn't say. There's this line from the movie Thin Red Line where there's a lieutenant captain, and the lieutenant says, "You're right, Captain," and the captain says, "You don't have to tell me I'm right. We'll just assume that." Speeman organizer is induced by an increase in Speeman would be ghostoid. See, I kind of like changed the order of this stuff to make it a little more challenging. So Speeman is the goosecoid. Oh, I'm sorry. No, it's the XNRs that are released from Newcoop center. This hormone released from Newcoop center is caused by the transcription factor CM1, in which itself was induced by dorsalizing factors that increased beta-catenin in the area opposite sperm entry point. Speeman is produced by the transcription factor goosecoid, and it releases, among other things, cordon. Okay? So normally I would say I would do it in a different order, so I kind of messed up the order because I'm that kind of guy. Okay.
I'm not sure if you want to show us the question, but I can't see the question.
Oh, you can't see it. All right. I will, for the next one, I have to change my share, I guess. I will do that for this one. Thanks for telling me, Mairead. Okay, match the experiment with its probable outcome. Use each term only once. Okay, cells from the dorsal, the experiment with its probable, use each term only once. Cells from the dorsal, oh, okay, so we're gonna culture cells from the dorsal margin of the blastula. This would be dorsal mesoderm. Vegetal cells allowed to develop alone in culture. That would be undifferentiated endoderm. Cells from the ventral margin of the blastula. That would give us ventral mesoderm. Animal cal cells placed on top of the vegetal tissue. That would become general mesoderm. Animal caps alone will be epidermis. Okay, so this is from that one series of experiments we talked about, which were like the early dissection and explant and transplant experiments that showed that there were diffusible factors that were causing these things to happen. So, you know, you had your blastula, you just take the animal cap off. What does that become? The margin on the dorsal side, the margin on the ventral side, the endoderm, all these different things separately. What do? And you take the animal cap and put it on the endoderm, and you get all kinds of mesoderm being formed there. So that was just general mesoderm. Okay. This one? Dorsal ventral axis, can you see this? Yes, okay. The dorsal ventral axis is made by the release of the hormone blocking agents from the Speeman organizer, including, now we've got cordon and noggin, so it could be either one. Let's put this one up here. Could also be Cerberus, but let's try this first. The role of these is to block BMP activity. In fact, one of these blockers, blank, shuttles the hormone to the ventral side. Ah, that's cordon. So now let's put noggin up there. Key to this is you can only use each one once. When the shuttling protein gets to the ventral side, the enzyme ADMP breaks it down, releasing the hormone that's being shuttled. The anterior-posterior axis is made by blocking the hormone Wnt, and that's being done by Cerberus. Okay? So again, being a little sneaky here, Cordon or noggin would be fine for the first one, but you can only use each one once, and you needed Cordon for that third one. So there we are. Okay, fate map time. Okay. So this will become the nervous system. Somites are middle mesoderm. Blood and heart is ventral mesoderm. Epidermis is ectoderm. Gut is there. And your dorsal mesoderm will include some notochord. Okay, again, this was just a figure from the class. Okay. Map signal transduction, okay. In the Wnt pathway, blank phosphorylates blank in the absence of Wnt. That would be GSK3 beta phosphorylates beta-catenin in the absence of Wnt. When Wnt is present, it binds to the frizzled, which is the receptor, which stops the phosphorylation. In the TGF-beta-like pathway, the hormone binds to the type 1 receptor, which then binds to the type 2 receptor. The receptors cross-phosphorylate and then phosphorylate the receptor SMAD. And this then binds to the cytoplasmic SMAD. And I gave you a couple extra ones there just to make your life more difficult. And I believe I got all these right. Woohoo! I'm getting an A in this class. Of course, I give myself extra bonus points every day just for showing up. You guys don't get that. Okay, how are ectoderm, mesoderm, and mesendoderm specified in the zebrafish blastula? So I think a lot of people didn't realize I was asking about signaling, but this was all about that signaling we talked about in the last class before the review. So this is all about nodal 1, nodal 2, and lefty being released by the yolk syncytial layer. So nodal 1 and 2 will both hit the first layer of ectoderm and turn that into mesendoderm. Nodal 2, I believe, drops off, and nodal 1 keeps going. And then that lower amount of nodal makes that next layer be mesoderm. And then lefty blocks nodals from going any further, so the top layer stays as ectoderm because it's got no nodal response at all. So that's what I was looking for there. Some people too added in, which I thought was really cool, that you get upregulation of brachyury, which then induces FGF, which induces brachyury, and that keeps the layers separate. I think that was what some people said. Okay, totipotent. This is another one. I think, again, probably the people who didn't come to class didn't know what I was talking about because they didn't really give a good answer for this. In Xenopus, after the first embryonic cleavage, each of the 2 cells is totipotent. After the second cleavage, each of the 4 cells is pluripotent, but not totipotent. Why is this, and what does it mean? So totipotent means it can become anything, and after the first cleavage, you get that cleavage that goes through the dorsal determinants. So each of those cells has got dorsal determinants on it. So they've got everything they need. After the next cleavage, it goes around this way. Now you've got 2 cells that don't have dorsal determinants and 2 cells that do. So the ones that do will form an embryo, but without gut because you're missing a lot of ventral structures. And the other side will turn into just undifferentiated ventral tissue because you don't have the dorsal determinants. So the pluripotent means it can become many things, but not everything. Okay. you have any questions, please just shout them out here. Okay. This is one that I thought was particularly clever question. An experiment is done with xenopus embryos to determine the importance of certain signaling pathways. Antisense mRNA is made for SMADs2 and 3 and injected into both cells of at the 2-cell stage. What would you predict to be the outcome of this experiment? So what you need to know to answer this is that nodals go through SMADs2 and 3. So you've got BMP signaling, but you don't have XNR signaling at all. So what are XNRs important for? Mesoderm induction, but you still have BMP there to get mesoderm. But more importantly, they're important for producing the Spearman organizer, which is dorsal mesoderm. So you will, you may or may not get the Newcoop center. What causes Newcoop center? It's Wnt and, oh God, listen to me, I'm getting stuck now. Wnt, which is beta catenin, and XNRs, I believe, together make that Newcoop center. Anyone back me up on this? What are the two factors that cause the upregulation of the Newcoop, or the production of the Newcoop center? Upregulation of CMOA. For a bonus point, anybody?
I think it's like Wnt and XNR, right?
Wnt and XNR, so I was right. Okay, that was Tyler, you will get a point. Okay, so if there are no XNRs, you probably won't get a Newcoop center, but you will definitely not get a Spearman organizer. So you're gonna get an embryo that has got ectoderm on top, some mesoderm in the middle, endoderm on the bottom, but it's not going to gastrulate because you don't have the organizer anymore. That's what you will probably end up with if you're knocking out all XNR signaling. Okay. And then this one, again, I thought this was an easy one, but a lot of people just plain didn't get it. And I think, again, it's the people who don't come to class and weren't listening to me prattle on about it. So in the figure below, animal caps are dissected away from embryos and exposed to beads soaked in varying concentration of activin. What does this experiment show? So this is all about showing that a concentration gradient give you patterning, right? That's what this is showing. So here you've got a low dose of activin, and brachiuria comes up, which is mesoderm in general, kind of mid mesoderm. Well, all mesoderm has got brachiuria. Then you use a high concentration, which means the higher concentration here, but lesser as you get further away. So here you're getting goosecoid coming up, being dorsal mesoderm, and then brachiuria here, and then these are unexposed, and so it stays ectoderm. So here you're getting a patterning based on concentration of activin. And this is the one I said earlier, and I was wrong. This is the one where some people were saying brachiuria upregulates FGF, which upregulates brachiuria, and that gives you a more distinct brachiuria expression. But also brachiuria inhibits goosecoid, and goosecoid inhibits brachiuria. So that gives you this distinct margin between cells that express one and cells that express the other. But that wasn't important for the answer. Mainly, I wanted you to tell me about concentration gradients producing patterning. That's what the experiment was about. Okay, most people did pretty well in this exam. Some people did not do so well, but you know, that's the nature of exams, I guess. It's so hard. This whole online stuff is hard. I know it is. If anyone needs more help, just please let me know, and I'll help you out on it. Okay. Any questions or comments about the exam? Okay, let's go ahead and learn some new stuff then. So we'll start out reviewing a little bit what we talked about on Monday. Today's Wednesday. Is it just me, or does it seem like the week starts and you work and you pick your head up from working and you're like, oh my God, it's Friday. What the hell happened? These weeks just seem to go by so fast.
That happens with me all the time.
What's that?
That happens with me all the time.
Yeah, it seems like the week goes by so fast, but the time in general goes by so slow. It's like we're still stuck in this COVID nonsense. But anyway, we're going to start with on slide 11 here, mainly because I just want to back up so that we're all in the same living space. So primitive streak. Primitive streak's all about your Kohler's sickle down here that starts to release FGF. And then you've got hypoblast, or Wnt actually. Wnt and FGF are causing the production of this nodal releasing cells here. And this is what's going to start the primitive streak. So you've got Wnt and FGF signaling, which is really important here. So it'll stay there until you get the hypoblast out of the way, because the hypoblast is releasing Cerberus, which is blocking the spread of that primitive streak. But then you've got your prospective endoderm that comes and replaces the hypoblast. And pushing the hypoblast out of the way is pushing the Cerberus further down this way, and then the primitive streak can start to move. Now, what the primitive streak actually is cells that are proliferating out here, they're dividing, and then they're coming down through this, this depression, the primitive streak itself. And in this primitive streak, they're being induced to become mesoderm and endoderm. And they're crawling inside. Again, these are mesenchymal cells, which means it's not a coherent sheet of tissue, it's individual cells crawling. And these come down through that primitive streak and start to make layers here. So as the primitive streak, expands up towards the anterior end, you end up with three layers of tissue there. You end up with your ectoderm on top, mesoderm in the middle, and endoderm on the bottom. When it gets all the way to the top of the, all the way to as far anterior as it's going to go, it stops, right? It doesn't go all the way to the anterior end, and that's kind of what we're going to be talking about today. It stops almost at the anterior end, and then it forms this Hensen's node. I got a call I got to answer here. Let me hang on for 1 second. Hello? Oh, can I call you right back? I'm in a class now. Oh, great. If I bring it in this afternoon, can I get it tomorrow afternoon? Is that possible? Excellent, thank you. Good news, people, I'm gonna get my computer fixed. I won't be dumping off on you like I did last week. Okay, what the hell was I talking about? Okay, so we don't get all the way to the anterior end, but it gets so far and it stops, and then it becomes this Hensen's node. That's where we're picking up the story today. What is Hensen? What's going to happen anterior to it, and what is the Hensen's node, and what is it doing? So this is your fate map at this part. Now remember, I talked about this Monday. Fate map doesn't mean it's this tissue yet, but at this stage, this is what these tissues are going to become. This particular fate map doesn't show what's underneath, it just shows the surface. So this surface ectoderm will all be neuroectoderm here. This is where the brain's going to come from. And then you've got epidermis, which will come over the top when you get the neurulation happening in the fold closing up. Now you've got some mesoderm behind it, which will become somites and lateral plate and blah, blah, blah. Okay. So this is what is interesting today. So here's your Hensen's node here. Cells during gastrulation and now at the start of neurulation that came through here are going to go laterally and anteriorly. And these are going to go underneath. These are going to go underneath what will become neuroectoderm and turn it into neuroectoderm. Okay, so if you look at this, okay, this is the gastrulation, neurulation, zipping up, zipping down, gastrulation, neurulation. Your head process is going to form out here. So at this point, neurulation has started, but you don't have any head process. So what happened here, those cells that went in went underneath that upper layer of ectodermis, and they make the brain and the head structures. So here's Hensen's node here. And the mesoderm that is kind of posterior to the head process becomes somites. We'll talk about somites probably in the next class. Some of it will become notochord. And then above it, here's that ectoderm that is now being turned into neuroectoderm by factors being released from this mesoderm here. So where am I now? Hang on. Slide 19. Let me look through my notes just to make sure I haven't left something out. Oh, just backing up a tad. So what I wanted to say was this is all laid down during gastrulation. And the interesting thing I read in the book about this mesoderm that's being induced during gastrulation, you're getting patterning here, both of your anterior posterior, obviously, as it's going forward, but you're also getting dorsal ventral patterning, which is like the depth of the embryo from the top down to the belly. Although you only have like 3 layers of cells here. So how can you get dorsal ventral where they're being patterned, but they're not that yet? So they've gone out, and they've had certain genes turned on that will make them turn into the ventral structures eventually. So especially the stuff that goes very lateral like this, the cells go in and some go sideways, some stay axial. The ones that are sideways will be ventral. So you're kind of looking at the dorsal ventral axis like this, dorsal ventral, going from axial to, or medial to lateral is dorsal ventral here. And again, it's not really dorsal ventral, but they've all been specified to become dorsal to ventral already at this point. Does that make sense? Hang on a second. I just want a little feedback, because sometimes I think I talk and nobody knows what I'm talking about. Does that make sense that they're patterned, but they're not patterned? Give me a thumbs up or a thumbs down. Thumbs up, thumbs up. You know, you have a little thing here, reactions off to the right. Can everyone give me a thumbs up, thumbs down, laugh, cry, whatever? Oh, Michelle's giving me a thumbs up. Okay, now I go over it one more. So Kwame wants me to do it again. Farouk's got it. Damn it, that Farouk's a smart kid. Okay, let me go over that again. Bum, ba, bum, 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 ba, bum. Okay, so where are we? So primitive streak-wise forms down here because of FGF and Wnt signaling, moves forward as that hypoblast goes away and it stops, and Cerberus is moving away too. That allows the primitive streak to come this way. What the primitive streak actually is this cells coming in and going sideways. So going in through the center and going lateral and anterior, like that. Now, as they come through, they're going to be specified to be either dorsal or ventral. The dorsal will stay axial and the ventral will go more lateral, okay? And again, it's not like you've got a dorsal ventral axis yet because you've only got 3 layers of tissue, but you will eventually have that after neurulation. And this mesoderm that has come through has been specified to become ventral, and it will kind of like wrap around and become the belly, the bottom part of the embryo. Okay, Kwame? Kwamster? Kwamarama? Okay. Okay. By the way, you guys can make fun of my name too. It ain't hard with a name like Slish. All right. Neurulation. Okay.
So, Dr. Slish, what did you mean by it's patterned but is not patterned at the same time?
I was being a jerk. I guess what I'm saying, it's specified, but it's not really patterned yet. Okay. So which means those cells will become ventral, the ones that are farthest out, but they're not really ventral yet because it's just flat. There's no up and down in yet, you know? That's what I mean. Does that make sense?
Yeah, now it makes sense. Yeah.
Okay, okay. Yeah. Sometimes I get jokey and I think I confuse people instead of making things clear, and that's clearly not my job. Okay, so head fold. Okay, so we did this head fold. Let's talk about it a little more. So as this is really going down, you've got mesoderm going forward and underneath this ectoderm, and that mesoderm, you can see here, is releasing factors that are going to make this into neural tissue. And that's how your head is being formed. Now, one of the factors being released here is kind of interesting. So you've got one of the things that's going to make this, that's being released from mesoderm, that will make this into neuroectoderm is FGF. Now, you may well ask, well, we've had FGF here the whole time, right? FGF and Wnt is what's making this primitive streak go forward. So why doesn't everything become neural because it sees FGF? Good question. Well, the reason is because until this node starts to regress, this ectoderm is not able to respond to FGF and become neural yet. And that's because the genes that will make this into neural are all boxed up in the nucleus, okay? So in the nuclei of the cell, one regulation, we always talk about genes being turned on and off based on a transcription factor hitting the upstream regulatory region and turning that gene on, right? Another regulation at the chromosomal level is the packaging of the chromosome. If the chromosome's all packaged up, then the transcription factor can't get to its regulatory element and the gene doesn't get turned on. And that's what's going on here. Some of these transcription factors that get turned on by FGF can't get to the neuro-inducing genes because they're all packaged up. Now, once you get into this post-gastrulation period, then you chaperone proteins pull the histones off the nucleosomes of the DNA, and now you get the DNA stretched out, so now it can actually be accessed by the transcription factors. So basically what I'm trying to say is before you reach this post-gastrulation stage, the ectodermal tissue is not competent to respond to FGF and become neural tissue. Once you get to this post-gastrulation stage, now it is. So FGF is going to help turn that ectoderm into neuroectoderm, and that's where your brain's going to come from. In addition to that FGF signaling, though, we also have BMP inhibition. So you have to inhibit BMP in order to get neural tissue. Kind of like the default is to become neural, but if you've got and BMP will cause that neuroectoderm to become epidermal. So we've got inhibition of BMP, which is caused by cordon and noggin that's being released by that Henson's node when it forms. Okay, so that's very similar to what we saw in Xenopus. BMP is ventral or epidermal, and you inhibit that with cordon and noggin, and you get the opposite, which is either dorsal or neuroectoderm. Okay, questions about that? Comments, discussion? Okay. I must be doing a good job today. Nobody's got any questions. Okay, Henson's node. Let's talk about Henson's node and the regression that happens during neurulation. So what is Henson's node? It's a depression in the ectoderm, and it is preceded by a stem zone. And the stem zone is where there's a lot of FGF and Wnt, and cells are dividing really quickly, and they're being induced to divide and to migrate. So they're coming in. So it's similar to gastrulation in that you've got ingression and moving anterior and lateral as that Henson's node is moving. The key thing to remember is in the stem zone, you've got neuromesodermal cells. So this is kind of similar to our mesendoderm, which is cells that are kind of endoderm and they're kind of mesoderm, except now they're kind of neuroectoderm and they're kind of mesoderm at the same time. Let me go to the next slide. Okay, and this is why we have that. Now this is a little bit complicated, but it's pretty important. So imagine this is the node, right? And the node is moving forward. Right here at kind of the, hang on a second, it's not really the leading edge, kind of the trailing edge of the node. Trailing edge of the node here, you've got this stem zone, and this is where cells are dividing and dividing and dividing. They're being induced by FGF and Wnt. Now, FGF, hang on a second. So these neuromesendoderm cells then can be either ectodermal or mesodermal cells. They're both expressing brachyury, right, which is a mesodermal marker, and SOX2, which is an ectodermal marker. We saw SOX2 once back in chapter 4 when we were using it as a neuroectoderm marker versus an epidermal ectoderm marker in an experiment way back in chapter 4. But just keep in mind, SOX2 is ectodermal. It's a transcription factor that's a marker for that tissue being ectodermal. So these cells coming through are expressing both mesodermal and ectodermal markers, and they will differentiate. If they stay in the middle, they'll become spinal cord. If they go out, they will become somites and nodal cord. So you're going to get a differentiation based on where they go as they come through. And we'll get more detail on that in the next class. But all I want to get across here is they're seeing both FGF and Wnt. FGF, as I just said, is a neuro inducer. So FGF is going to upregulate a transcription factor called Churchill. Again, stupid name. Who knows why they call it Churchill, but it is. So you got to memorize the fact that Churchill is a transcription factor, and it specifies, it specifies that those cells will become neuro. Wnt is going to upregulate more brachyury. So cells that end up with more Wnt will become mesodermal. Now they're going to be either axial, which will be nodal cord, or paraaxial, which is somites. Right? Axial in the center, right underneath the neuro, and then paraaxial next to it, somite. And so basically this node moves, right? And so as it moves, it's kind of like a wave of inducing activity. So you've got this, on top of, you've got these epiblast cells on top, and as that node comes down, they divide and then they go in, they become something else and they go in and spread out. And that's a wave of that activity going from anterior to posterior. And that wave is basically a signaling wave. And the signaling, you've got two different signaling things that are going on here. From that node, you've got a lot of Wnt and FGF being expressed, right? That's causing them to divide and to move. But at the back end of that, you've got somites releasing retinoic acid, new signaling molecule we're going to talk about. So if we forget about retinoic acid for a moment, these cells come in, and as they're coming in, they're responding to FGF and Wnt, and they're producing FGF and Wnt, and they come through. Now, as they go through, they stop releasing FGF and Wnt. So you can imagine them coming through, spreading out, and as the node regresses, they're seeing less and less FGF and Wnt, right? Because that's concentrated in the node as it's moving. So these cells respond to it, crawl through, and they sit there and wait, or sit there, move into position, and then FGF and Wnt drops off, and they start to differentiate. And another factor that's going to be involved here is this retinoic acid retinoic acid signaling molecule. So retinoic acid is kind of like a steroid hormone. It's a small molecule and it is lipophilic. So it's got an internal receptor. So retinoic acid will be made in cells of the somites at the back end of that node, right? So that's going to limit FGF and Wnt. It's going to stop the Wnt signal because it's making retinoic acid. Retinoic acid comes out, goes into the next door cell, binds to a receptor, and turns on genes there. So you've got this gradient of FGF and Wnt here because of the movement of the node. So the source of FGF and Wnt is going away, but also because as it gets further away, it starts to differentiate, it becomes a somite, and it releases retinoic acid, which is blocking the FGF Wnt signal. So you've got a gradient of these two different things, and they inhibit each other, which increases that gradient. So if we go here to slide 24, you can see FGF as is produced will move forward and will block production of retinoic acid by inhibiting the production of the enzyme that makes retinoic acid. So when you're close to the node, there's no retinoic acid because there's high FGF. It's also upregulating this enzyme, which breaks down retinoic acid. So right near the node where FGF is high, you're getting a blocking of any retinoic acid. As you get further away from that node, retinoic acid starts to show up and it blocks the production of FGF. So that's what's giving us this double gradient kind of, and really, and the big difference here between them, is that FGF and Wnt are involved in proliferation, division of the cells, and crawling. And retinoic acid is going to be involved in differentiation. So they're kind of, as they're coming through their neuromesodermal, as they get past that node, they become neuro or mesodermal, and they start going down their respective fate maps. Okay. And that's about all I wanted to talk about today. We'll talk about this next time. So I want to stop here for a second and ask, how did everyone hang into that last discussion? Because it's kind of complicated, I think, anyway. Questions, comments? Yusuf, are you hanging in there? He's not even paying attention.
Wait, sorry, what'd you say?
I just said, are you hanging in there?
Yeah.
Okay. I knew you would be. I just wanted to tease you. Okay, so Elijah says, did you say somites produce retinoic acid? Yes. So the cells come through and eventually they differentiate into somites and they release retinoic acid. Okay, any other questions? Comments here? We still have 6 minutes, so I'm going to sit here and stare at you for the next 6 minutes, unless you have a question. No questions? Okay. Well, I guess Friday we'll have a little quiz to start off on to make sure everyone's up and running. Exam on Monday, right?
Wait, what?
So you said what?
See, you are listening. I just wanted to see if you're listening. Okay, so we'll meet back here on Friday, same time, same station, and we will talk about somites being formed, I think. We'll see. All right. Have a good day, all.